SciAnts. We get experimental SciAnts. We're curious, non-judgmental.
Do y'all want to know the new species? It is, in fact, a bird. These birds are toxic. They're one of 21 species of bird that have toxicity. These are in Papua New Guinea. This is the bird. It's actually one of 2 species that they discovered, both of which in Papua New Guinea were discovered around the same time. They are resistant to the same toxin. It's a neurotoxin, and it's called batrachotoxin. I have this written out because I'm not gonna remember how to say this. Brand new species. It is toxic with a neurotoxin. It is not poisonous because it does not create its own toxin. So that is the difference. If it were poisonous, it would be making it itself, but it is not making the toxin itself. It is toxic. It is getting it from its diet. So it is, again, called batrachotoxin. It is a neurotoxin. And so, yeah, if you pet it, penguin, there should be no issue with these birds. They discovered 2 of them, and the reason this is surprising, because in the last 20 years, no toxic birds have been discovered. It was not clear in the paper of why why the large gap, but it's just something to keep in mind of well, it's really rare, and I wonder why it's so rare. Monarch butterflies also acquire toxicity from eating the milkweed diet, but the closest thing actually to how it functions is poison dart frogs. So poison dart frogs, actually discovered in Papua New Guinea, the same location as these birds, have the exact same toxic defense, which is kind of neat. So 2 animals have independently evolved to be resistant to a particular toxin, so it allows the toxin to build up in their system. And actually what's really cool about this paper, remember how the other new species papers that we've talked about are all here's morphology, here is what it looks like, right? Here is the species, here is the genome sequence, and we're done. This paper actually went to identify the molecular mechanism that makes these animals resistant to the toxin. So it is, in my mind, a tier above our usual different species papers, because not only did they go in and be hey, this is a brand new animal, but I'm also going to try to tell you what the mechanism of why they are able to survive on this toxic component is. Here is the gene, though, that was discovered. So it's a sodium voltage gated channel, meaning it's involved in neuronal signaling. So neuronal firing, this gene is involved for. It maintains the balance of sodium and potassium ions, gradient of those, allowing neurons to fire. What's mutated about it is the area in which the toxin would normally bind. So what you're finding is, and I think this is really cool, the toxin is unable to bind to these receptors, and that's what makes the birds resistant, because the molecule can go in their system, it just can't do anything. Versus if you were to put this molecule, the toxin, onto human cells, onto most other bird cells, onto insect cells, it would shut down that gated channel. So that's why it's a neurotoxin, because it acts on this particular voltage gated channel. And the reason these 2 birds are resistant is because the area of the protein that the toxin would bind to looks different. And so the toxin can't bind. So it flows instead in the system. Does the toxin have to get past the blood-brain barrier? No. So where it's acting it can act on just neurons, right? So it can end up crossing the blood-brain barrier to actually do it. It's a small enough molecule to cross that barrier, but it doesn't need to, because your neurons are open access, right? So in the system where it crosses a bloodstream and goes in, it can attach to neurons and start shutting down different parts of the body. And eventually it can get into the brain and cause even bigger shutdown. So it will eventually, at high enough concentrations, lead to death, not because it's paralyzed your neurons in your arms, but it will go into the brain and it will just stop, the brain will just stop firing, because what you're doing is you're disrupting that sodium voltage channel. So right, that is a really, really cool adaptation that these birds have, and it's very elegant. And what's really cool is the poison dart frog, also in Papua New Guinea, that's also resistant to this toxin, also has a mutation in this gene. Now the location of the mutation is different, so it is not the same mutation between the 2 animals, but the gene that's mutated is the same. Does that make sense, y'all? And I think that's a neat component of it. Can the neurotoxins reproduce, or the animal limit to what it consumes. So great question. If the animal is not ingesting the diet that confers the toxicity, it is not toxic. So it will wear off with time, meaning that if Penguin wanted one of these birds, so it is the rufous-naped whistler. Here is the bird, if anyone's interested, one of this bird species. If you bring it home to the United States, presumably the diet that you would feed it is different. It can survive off other food. It doesn't just have to be the native food. Those won't have the neurotoxins, and it will not be toxic. The new species, and here's the other one. This is the image that we started off the stream with, the Regent Whistler. And again, they're really pretty birds. Granted, I'm a sucker for a lot of birds, so like this is me saying they're cute means nothing, because I will probably say most birds are cute, but it is just a really pretty bird. What they do say in this paper, and I checked the citation as well, is that its current limited knowledge suggests the birds acquired the BTX toxin via ingestion of the toxic beetles. So we know the beetles are toxic, and it is not confirmed that they haven't like proven that that's the diet source of them, right? So the only way to actually prove it would be either to bring everything that it might be able to eat into a lab and ask, and then remove the beetles, or genetically engineer a beetle that isn't toxic, and then again you have to do a similar kind of experiment on that front. So I am not comfortable answering if the beetles are toxic or poisonous. And I say that because the paper about the beetles just said that they're toxic. They didn't prove where or what the beetles were getting it from. Like if the beetles are producing themselves or it's something else in the diet. So I didn't dig further. Okay, you get how you evolved to be poisonous yourself, but how do you evolve with the poisonous symbionts? So what it was in this particular one is that both birds have this gene, this SCN4A gene. That is a component of your nervous system, and it is to maintain the sodium-potassium levels between neurons on the outside of the cell, because it is the change in that voltage that fires a neuron. What the toxin does is it binds to these channels, opens them up, and just essentially shuts down neural function. But these birds have evolved mutations in the binding area of the toxin, where the toxin will usually bind to shut down the receptor. That area is mutated, so the toxin can't bind anymore. So instead, the birds are fine. The toxin is free floating in their blood. And then if anyone eats it, then they die. So it most likely is. One had a question earlier tonight in chat, and this is essentially what ended up happening, right? In a not as crude way, possibly, but it was probably some bird was like, hey, what is this? And ate a beetle and maybe didn't get sick from it, right? And so that bird that didn't get sick from eating the beetle might reproduce better than the birds that do get sick from eating the beetle, because it has this extra food source that most other birds do not have. So there's less competition for this particular kind of food, so it can do better. So its genes are more likely to be passed on. And so that's most likely the situation that ended up happening. Does that make sense? There is a total of 21 non-toxic birds in this family that they've identified. So in the family, there's 21 birds that are not toxic, and then there were 6 previously that were toxic birds. And now there's an additional 2 identified. How does their toxicity present? Like, use it to kill prey. So for these birds, they don't use it to kill prey. It is a defense mechanism, so that if something else tries to eat them, that thing will die. And then they learn, those predators learn to avoid these birds. Same thing as poison dart frogs. Here we go. This is a neat component as well. So this was my favorite figure of the paper, because this is not something that you're standard seeing within a lot of these animals. But here you're seeing each of these. This is the whole receptor, the voltage gated channel. You see it's transmembrane, meaning that it goes from the inside of the cell to the outside of the cell, back inside multiple times. And then in blue are where the poison dart frogs have changes in their sequences. And then in red are where the birds do. So while it's the same gene that's mutated in these 2 animals, and it's the same toxin in the 2 animals, the BTX, you can actually outline where the mutations are within the protein. And those mutations are in 2 different spots in the 2 animals. And so it seems like you can mutate the protein in these different locations to confer resistance to this BTX neurotoxin, which I think is really cool. And again, this comparative genomics approach is really neat. And again, here in red are the toxic birds, and they're demonstrating where there's mutational changes within these components. Like here, this Pachycephala, this entire sequence is missing. In this sequence here, instead they have an N instead of a D. And so you can look at like where the amino acid changes are that changes the protein and confers that resistance to these animals. And again, it's just really beautiful. Like they found out the molecular mechanism. Not only did they identify, yes, this is a different bird, but we look at the actual structure of the protein and identify why it's different and where it's different, especially between two animals that use the same thing. They further went through and actually identified from the blood of the bird that there was a present peak in the mass spectrometer of the particular toxin. So they showed that it was actually in the bloodstream of the bird. So that was the big thing, I think, in this supplement that I was excited about seeing, is that they did do the blood confirmation between the two. They had the genome collections published as well. They did the genetics, they did the measurements. And again, the big one was identifying what compound was actually mutated, like what component of the genome was different. Alrighty, y'all. So that was our first science news story of the evening, was this two new species of bird that was identified that are toxic. And we talked about why they're toxic, which is part of their diet. We talked about the molecular mechanism by which they're toxic, and the comparative biology between them and poison dart frogs. We also looked at some other toxic birds as well, and have come to the conclusion that they don't make their own toxic components, but rather they get them from their diets. That's the common theme behind it.